Welcome, Zoo Assembliers! My name is Zouk Azalishvili and I'm the founder of Zoo Assembly. Zoo Assembly is an online podcast for the highest yield basic science and clinical knowledge tested on USMLE Step 1 and USMLE Step 2 CK. The information discussed in this podcast is intended only for educational purposes. It's not intended to prevent, diagnose, or to treat the medical conditions in real clinical practice, nor is it intended to reflect the policy and the guidelines of various health institutions. Simply put, we serve you to butcher your step exams. Please subscribe to our podcast, Facebook, Instagram pages, and the YouTube channels down below in the description of this episode so that we keep you tuned for the news at Zoo Assembly. Now, let's start rolling. We are continuing our musculoskeletal series, and in today's episode, the first topic that we are going to discuss is the types of the skeletal muscle fibers. There are two types of skeletal muscle fibers. We have type 1 and type 2 fibers. We need to compare and contrast these type of fibers, and we need to explain what type of activity increases the proportion of each of these types of fibers in the muscles. Okay, let's start by discussing the type 1 fiber, then we'll discuss the type 2 fiber, and then we'll compare and contrast these two. Type 1 fiber is the slow contraction fiber. In other words, so the type 1 muscle fibers or muscle cells contract slowly. And type 1 fibers are red. Guys, do you know the reason why type 1 muscle fibers are red in color? Abso-freaking-lutely. The reason for this is that type 1 fibers have the high content of the myoglobin. And myoglobin, let me remind you, is the protein which stores the oxygen in the muscle cells for the oxygen to be used in the oxidative phosphorylation later on. And the myoglobin and the heme group and the iron there confer the type 1 muscle fibers the red color. The main metabolism for type 1 fibers is the oxidative phosphorylation. We know that we produce ATP either through anaerobic glycolysis, which is always the first pathway until the oxidative phosphorylation, and then we can increase the ATP production by utilizing the oxidative phosphorylation pathway in the mitochondria. And since we mentioned that type 1 fibers mostly depend on oxidative phosphorylation, then it's logical that type 1 fibers should have the high content of mitochondria as well. So to summarize, the type 1 fibers have high content of both myoglobin and the mitochondria. And since they depend largely on the oxidative phosphorylation rather than the anaerobic glycolysis, they can produce sustained contraction. The reason for this is that type 1 fibers produce ATP in the stable, sustained manner, and therefore they allow the whole muscle to stay in contracted position for a considerable amount of time. This is why the type 1 muscle fibers are especially helpful for the endurance training. And endurance training increases the proportion of type 1 muscle fibers in our muscles. The way we can remember the different characteristics of type 1 fibers is by the mnemonic of the first aid. This is a very famous mnemonic which I always use, and I would recommend to you that you use it as well. So the mnemonic sounds like this: I slow red ox. I depicts that we are talking about the type 1 muscle fibers, right? Because the Roman numeral 1 looks like the Latin I. Slow reminds us that type 1 fibers have the slow contraction velocity. The word red in the mnemonic reminds us that the type 1 muscle fibers are red because of the high myoglobin content. And finally, ox reminds us that type 1 fibers depend largely on the oxidative phosphorylation. Okay, now that we've discussed the type 1 fibers, let's move on to the type 2 muscle fibers. Type 2 muscle fibers are the fast contraction fibers. In other words, they contract very fast. And at the same time, type 2 muscle fibers are white. Based on our discussion on the color of the muscle fiber and the myoglobin content of that muscle fiber, could you please tell me whether the type 2 muscle fiber has high or low myoglobin content? Absolutely. The type 2 muscle fibers have low myoglobin content, and this is why they are white. Myoglobin, as we already mentioned, is the protein that stores oxygen inside the muscles for the oxidative phosphorylation. Now let's think about this. If the type 2 muscle fibers have low content of myoglobin, then they have low content of the oxygen, which means that they cannot efficiently run the oxidative phosphorylation. Because oxidative phosphorylation always needs the oxygen as the final receptor of the electrons. So since the type 2 fibers have low myoglobin content, they depend largely on the anaerobic glycolysis rather than the oxidative phosphorylation. And another reason for why the type 2 fibers depend mostly on the anaerobic glycolysis is that they have the low mitochondria content. As we know from the biochemistry, mitochondrion is the organelle which runs the oxidative phosphorylation on its inner membrane. So less mitochondria means less oxidative phosphorylation and more anaerobic glycolysis. We've already said that type 2 muscle fibers are the fast contraction fibers. Therefore, they are particularly helpful for the activities, the physical activities which require fast, non-sustained muscle contraction. Could you please tell me what kind of physical activities make use of this non-sustained fast muscle contraction? I really hope that you're saying that the weight training, also known as the resistance training, is totally based on these fast, non-sustained muscle contractions. Sprinting is another example. So if the patient, not the patient, but if the person just works out with the resistance training or sprinting, then she or he will have high proportion of type 2 muscle fibers in her or his muscles. And the way we can remember the different characteristics of the type 2 muscle fibers is by the first aid mnemonic again. And this is the relatively new mnemonic, but I like it very much. So it says 2 fast white antelopes. 2 reminds us that we're talking about the type 2 fibers. Fast tells us that type 2 fibers are fast contraction fibers. White reminds us that type 2 fibers are white due to low myoglobin content. And finally, N, so the first 2 letters of the word antelopes, remind us that the type 2 fibers depend largely on the anaerobic glycolysis rather than the oxidative phosphorylation. Okay, this was our discussion about the types of the skeletal muscle fibers. Let's move on to the skeletal muscle adaptations. Here, in this subsection, we'll talk about the atrophy and hypertrophy. Atrophy and hypertrophy are the cellular adaptations, and we'll touch on them in the pathology section as well. However, since we're talking about the muscles, it's worth discussing atrophy and hypertrophy here too. we need to think of atrophy and hypertrophy of the muscle in terms of what happens to the myofibrils and what happens to the myonuclei. Before we go any deeper, let me ask you a question. Zeus Emeliers, could you please remind me from the previous musculoskeletal episode what the myofibril is? Are you saying that the myofibril is the parallel, not the parallel, sorry, but the stack of sarcomeres in series? If you are, that's totally correct. So myofibril is just the stack of sarcomeres in series. And several myofibrils fill up the cytoplasm of each muscle cell. Well, myonuclei literally means muscle nuclei, or the nuclei of the muscle cells. In the setting of the atrophy, the number of the myofibrils decreases. And it's very, very high to know that the mechanism by which the muscle fiber or the muscle cell is able to remove the myofibrils is the ubiquitin proteasome system. Let me remind you that the ubiquitin is this tagging protein which binds to the lysine residues of the different proteins in the cell, and then ubiquitin brings that obsolete protein to the proteasome. Proteasome is a specific structure which chops down or breaks down those useless or obsolete proteins. And myofibrils, which are the stack of the sarcomeres, are composed of the proteins. So since the proteins composing the myofibril are breaking down, then the myofibrils are shortening and they are also decreasing in number. Additionally, atrophy is characterized by the decreased number of the myonuclei. Let's remind ourselves that each muscle cell contains several muscle nuclei along the plasma membrane. This is because the center of the muscle cell is occupied by the myofibrils. And then these muscle nuclei get broken down by the processes called pycnosis, karyorrhexis, and karyolysis. Again, these are the pathology concepts. However, there's no way that we can touch them here as well. So pycnosis means the shrinkage of the nucleus. Karyorrhexis means just breaking down the nucleus into multiple small fragments. And then karyolysis means the fading or actual disappearance of those nuclear fragments. Now that we've discussed the changes in myofibrils and the myonuclei in the setting of the atrophy, let's move on to the hypertrophy. Hypertrophy is characterized by increased number of the myofibrils. And this is a very high-yield concept. The muscle, the skeletal muscle, and generally the striated muscle as well, so I mean cardiac muscle, they are permanent tissues. In other words, they have a very, very small, minuscule regenerative capacity. In other words, the cells in the striated muscles cannot divide by mitosis to produce the similar cell. And when we say that the muscle is hypertrophied, we mean that the size of each cell is increased. It's not the new cells, new muscle cells that have been produced by hypertrophy. That's not true. Hypertrophy means that we have the same number of the cells, but each cell has increased in size. The way the muscle cells increase in size is that myofibrils are added in parallel. So sarcomeres, the stack of the sarcomeres are added in parallel, and therefore we get much more myofibrils inside each muscle cell. Additionally, hypertrophy is characterized by increased number of the myonuclei. The mechanism for this is that satellite cells interspersed among the muscle cells fuse, and they produce like multi-nucleated cell. I know that I've told you that muscle cells have little regenerative capacity, but I have not said that they have zero regenerative capacity. There are still satellite cells which act as the stem cells or regenerative cells of the muscles, so they can still fuse and produce the new muscle cells, but this is a very minor process compared to the increased size of already existing cells. Okay, we have now discussed the skeletal muscle adaptations, specifically atrophy and hypertrophy, and now we'll move on to vascular smooth muscle contraction and relaxation mechanisms. The smooth muscle cells are present in many internal organs and the sheaths of many internal organs, especially the GI system and respiratory organs as well. However, here we will focus on the vascular smooth muscle, that is, the smooth muscle around the vessels, particularly the arterioles. Vascular smooth muscle has a very important function of controlling the vascular tone. Vascular tone represents the overall degree of vasoconstriction, right? So that if the vascular smooth muscle contracts, then it just narrows down the vascular lumen and it increases the total peripheral resistance. In contrast, if the vascular smooth muscle relaxes, that will open up the arterioles and that will decrease the total peripheral resistance. Now we are going to discuss how the vascular smooth muscle gets contracted or relaxed. So first, there is some sort of stimulus which works on the endothelial cells. This stimulus, or agonist in this case, can be acetylcholine or bradykinin. And when acetylcholine or the bradykinin binds to their respective receptors on the endothelial cells, this induces the calcium influx inside the endothelial cells. Now, calcium in the endothelial cells has a very special function of activating the nitric oxide synthase, shortly NO synthase. As the name implies, this enzyme synthesizes the nitric oxide. Now, NO synthase takes L-arginine inside the endothelial cells and it cleaves the arginine into citrulline and nitric oxide. The nitric oxide received from this biochemical reaction then diffuses from the endothelial cells to the smooth muscle cells around the vessels. Nitric oxide, once inside the smooth muscle cell, activates the enzyme called guanylate cyclase. Guanylate cyclase exists in two forms. We have the intracellular form of this enzyme, which is inside the cytosol of the cell, and we also have the plasma membrane, so transmembrane protein, which is carrying the function of guanylate cyclase. Nitric oxide activates the intracellular isoform of guanylate cyclase, the one that's inside the cytosol. Guanylate cyclase converts GTP into cGMP. Now, cGMP activates protein kinase G, and then protein kinase G phosphorylates and activates the enzyme called myosin light chain phosphatase, shortly known as MLCP. As the name implies, myosin light chain phosphatase removes the phosphate group from the myosin head or myosin light chain. From the previous episode, we know that in order for the cross bridge and the contraction to happen, the myosin head should be physically connected to the inorganic phosphate. So if myosin light chain phosphatase removes the inorganic phosphate from the myosin head, then the myosin head can no longer bind to actin, and if there's no cross bridge between myosin and actin, there will be no contraction. In other words, activation of myosin light chain phosphatase through nitric oxide causes muscle relaxation. The way you can remember that nitric oxide causes muscle relaxation is that both of these words contain the letter X. Nitric oxide causes muscle relaxation. And this mechanism that we described right now is very important, and it underlies the pharmacological mechanism of the nitrate drugs. In pharmacology, we will talk about the nitrates like nitroglycerin, isosorbide dinitrate, isosorbide mononitrate, and there I will remind you that every drug, every nitrate drug, is the donor of the nitric oxide. In other words, once they get into our blood, they release the nitric oxide. After this, the nitric oxide diffuses into the vascular smooth muscle, and it performs all of those biochemical reactions that we've discussed several minutes ago. Okay, now that we have shown, now that we have discussed how the muscle gets relaxed, let's talk about how the vascular smooth muscle undergoes contraction. The neural stimulation causes depolarization of the vascular smooth muscle, and for all intents and purposes, any smooth muscle. The depolarization of the smooth muscle cell opens up the L-type voltage-gated calcium channel on the plasma membrane of the smooth muscle cell. This channel is called voltage-gated because it depends on the change in the voltage across the plasma membrane. And the depolarization means that the net charge inside the cell is positive while the net charge outside the cell is negative. And this change opens up the L-type voltage-gated calcium channels. Opening up the calcium channels on the plasma membrane of the smooth muscle causes the influx of calcium from the interstitial fluid into the smooth muscle cytosol. The calcium there binds to the protein called calmodulin. Calmodulin is called so because it's calcium modulated protein. So it's modulated and it changes its conformation when calcium binds to it. And the calcium calmodulin complex activates the enzyme called myosin light chain kinase, shortly MLCK. We have already discussed what MLCP does, so could you please tell me what the myosin light chain kinase would do? Absolutely. As the name implies, myosin light chain kinase phosphorylates the myosin light chain. In other words, it puts the inorganic phosphate group onto the myosin head. So once the phosphate is on the myosin head, then the myosin head can form the cross bridge with the actin filaments. And once we have the cross bridge, then we have the power stroke and the muscle contraction. So now you can see that calcium inside the muscle mediates muscle contraction. And that's true not only for the smooth muscle, but also for the striated muscle. The general rule of thumb here is that calcium equals to contraction. Every time the calcium content increases inside the excitable tissue, like striated muscle or smooth muscle, the muscle will contract. Okay, we have discussed the vascular smooth muscle contraction and relaxation here, and now it's time for us to talk about the muscle proprioceptors. Here we'll discuss the muscle stretch receptors and the associated reflexes, and the Golgi tendon organ along with its associated reflexes. The muscle proprioceptors include the Golgi tendon organs and the muscle spindles. The Golgi tendon organs detect the muscle tension, while the muscle spindles detect the muscle stretch or increase in the muscle length. Let's first talk about the muscle stretch receptors or the muscle spindles. And now we'll talk about the reflex arc, which starts from the muscle spindle. So whenever we stretch the muscle, this activates the muscle spindle fibers. And muscle spindle fibers are innervated by type 1A and type 2 nerve fibers. So type 1A and type 2 nerve fibers transmit the information about stretching to the dorsal root ganglion. The dorsal root ganglion, in fact, contains the cell bodies for those type 1A and type 2 nerve fibers. And then from the dorsal root ganglion, the information goes into the posterior horn of the spinal cord through the dorsal root. From the posterior horn of the spinal cord, some fibers go into the alpha motor neurons and some fibers go towards the inhibitory interneurons. So here's what happens. There is activation of the alpha motor neuron innervating the agonist muscle, and there is activation of inhibitory interneuron, which innervates the alpha motor neuron of the antagonist muscle. Let's take an example, and I think it will be much easier to interpret and imagine what's happening. Let's take the patellar reflex. We hit the reflex hammer on the patellar tendon, right? Hitting the reflex hammer on the patellar tendon will stretch the patellar tendon and the quadriceps femoris muscle, which is present in the anterior thigh compartment. Stretching the quadriceps femoris muscle will stretch the muscle spindles in the body or in the main part of the quadriceps femoris muscle. So this will relay the information through the type 1A and type 2 nerve fibers and the dorsal ganglion, to the posterior horn of the spinal cord. Now, here is what happens. There is activation of the alpha motor neuron, which innervates the quadriceps femoris muscle, the fibers in the quadriceps femoris muscle. Therefore, the quadriceps femoris muscle will contract, and this will prevent any more stretching of the muscle. Additionally, there is activation of the inhibitory interneuron, which finally synapses on the alpha motor neuron that innervates the hamstring muscles. The hamstring muscles, as we already know, are located in the posterior thigh compartment, and they normally are responsible for the knee flexion and hip extension. So they are antagonist muscles for the quadriceps femoris muscle. So if we activate the inhibitory interneuron, it will inhibit the alpha motor neuron, which normally innervates the hamstring muscles. Therefore, the hamstring muscles will be relaxed. And this activation of the agonist muscle and inhibition of the antagonist muscle will result in contraction of the agonist muscle. And inhibition of the antagonist muscle will prevent the overstretching of the muscle. So again, we already mentioned that the stretch response of the muscle is transmitted to the spinal cord through the type 1A and type 2 nerve fibers. And the muscle spindle itself is located in the body of the muscle. And the last thing that I'd like to tell you about the muscle stretch receptors is that all the deep tendon reflexes that we clinically check are mediated by this reflex arc. So first the information goes into the spinal cord, and there we have the polysynaptic reflex. So one fiber, let's say, synapses on the alpha motor neuron of the agonist muscle, while another fiber synapses on the inhibitory interneuron. And then the inhibitory interneuron again synapses on the alpha motor neuron of the antagonist muscle. So here you can see that we have three different synapses. That's why we call these reflexes the polysynaptic reflexes, or the reflexes which involve multiple neuronal synapses. Right, let's move on to the Golgi tendon organ. Golgi tendon organs are called so because they are located in the muscle tendons. In contrast, the muscle spindles are located in the body of the muscle. And the Golgi tendon organs respond to the muscle tension, not stretch. So again, muscle spindle is activated by the muscle stretch. Spindle stretch, right? SS. While Golgi tendon organ is activated by muscle tension. So tendon and tension. This is the easy way to remember what activates each of these muscle proprioceptors. And I have one question for you guys. When do we have the muscle tension? Well, we have muscle tension when we perform the contraction of that muscle, especially when we do the isometric muscle contraction. For example, lifting the weight requires the isometric muscle contraction, and this is what can activate the Golgi tendon organs. Once we have the tension, and once the tension is felt by the Golgi tendon organ, this will relay the information through the type 1b nerve fibers to the dorsal root ganglion. Again, dorsal root ganglion contains the cell bodies for the type 1b nerve fibers. The fiber exiting the dorsal root ganglion will enter the posterior horn of the spinal cord, and there the fiber will synapse on the inhibitory interneuron. This inhibitory interneuron normally inhibits the alpha motor neuron that innervates the agonist muscle, the muscle that experiences the tension. So if the fiber coming out from the dorsal root ganglion activates the inhibitory interneuron, then this inhibitory interneuron itself will inhibit the alpha motor neuron that innervates the tense muscle. If we inhibit the alpha motor neuron, this will stop transmission of any action potential to the appropriate muscle, and the muscle will stop contracting. This is how we'll prevent muscle from tensing too much, from tensing excessively. Again, as we already mentioned, the Golgi tendon organs are located in the tendons, and they relay their information through the type 1b nerve fibers. And the main stimulus for the activation of Golgi tendon organs is the increased muscle tension. Right, so we have now talked about the muscle proprioceptors, and now let's move on to the bone formation. Here we'll talk about the endochondral ossification and the membranous ossification. Again, there are two processes by which the bone is formed in the fetus, or in, I should say, the embryo. The first one is endochondral ossification. Let's talk about this one. And first, let's explain why it is called the endochondral ossification. At first, the body makes the cartilaginous model of the bone. So it's the model for the bone, but it's actually the cartilage, and it's made by the chondrocytes. But then the osteoclasts and osteoblasts infiltrate that cartilaginous model of the bone, and they replace the cartilaginous model with the actual bone. So this is why it's called endochondral. Chondros means cartilage, right? Endo means inside. So it means that first we have the cartilaginous model, and then osteoblasts and osteoclasts start to form the bone of the same shape from the inside of the cartilage. When the bone is formed, there are two types of bone. There is a woven bone and the lamellar bone. Woven bone is the immature disorganized type of bone. We all know that the major component of bone is the type 1 collagen. And in the mature bone, which is called the lamellar bone, the collagen fibers and fibrils are organized in a specific manner. So there are specific lamellae, or the layers, concentric layers of the type 1 collagen, and the collagen fibers in each layer are located perpendicular to the collagen fibers in another layer. To say it simply, lamellar bone is an organized bone, organized into the concentric layers of bone. But the woven bone is disorganized, so the collagen fibers don't have any spatial organization pattern. So they are just dispersed all over around. And woven bone is what is made after the fracture. For example, when the person fractures her or his bone, we know that the bone has the ability to regenerate itself. And the first bone that forms in the place of the fracture is the woven bone. Sorry. And then the woven bone is replaced by the lamellar bone time by time. Another very high yield condition in which the adult patients have the woven bone is, could you please complete the sentence? That's Paget disease of the bone. We'll definitely talk about Paget disease in a great detail in the musculoskeletal series, but from this point on, I'd like you to remember that Paget disease of the bone is characterized by this mosaic pattern of the woven bone. Okay, and the same thing happens in the endochondral ossification that we have already described in case of fracture. When the osteoblasts and osteoclasts first make the bone in this cartilaginous model of the bone, they make the woven bone. But then this woven bone is replaced by the lamellar bone. We should also know which bones are formed by the endochondral ossification. And the easy way to remember this is that almost all bones in our body are formed by the endochondral ossification. Specifically, these are the bones of the axial skeleton. Here we mean the vertebral column, pelvic girdle, sternum, and the ribs. Appendicular skeleton is completely formed by the endochondral ossification, and very, very importantly, the base of the skull is also formed by the endochondral ossification. The reason I'm emphasizing that the skull base is formed by the endochondral ossification is that some of us erroneously think that the whole skull is formed by the membranous ossification, but that's not true. Skull base is still formed by the endochondral ossification. Now that we've touched on the membranous ossification, let's move on to the membranous ossification. In membranous ossification, there is no preformed cartilaginous model of the bone. In this case, the osteoblasts and osteoclasts form the woven bone without the previous cartilaginous model of the bone. And later, this woven bone is remodeled to the lamellar bone. The bones which are formed by the membranous ossification include the bones of the calvarium, or the upper skull, which involves the frontal bone, parietal bones, and the occipital bone. The facial bones are also formed by the membranous ossification, and finally the clavicle is formed by the membranous ossification. In fact, the clavicle is the first bone to ossify. And the easy way to remember which bones are formed by the membranous and endochondral ossification is this one. Try to remember the bones which are formed by the membranous ossification. So these are the bones of calvarium, facial bones, and clavicle. Anything else, any other bone, is formed by the endochondral ossification. Okay, Now we've talked about the bone formation, and now let's talk about the cell biology of the bone. Okay, so cell biology of the bone will be our last topic for our today's episode. Let's start by discussing the osteoblasts. Osteoblasts are the cells which build up the bone. The way you can remember this is that osteoblasts build up the bone. So B in osteoblasts stands for builds the bone. The way osteoblast builds up the bone is that it produces the collagen. We already mentioned that the main component of bone is type 1 collagen. And the type 1 collagen is produced by the osteoblast. So after the osteoblast releases the type 1 procollagen into the interstitial space of the bone, then it also releases the bone subtype of the alkaline phosphatase. Alkaline phosphatase creates the alkaline environment in the bone matrix. In other words, it just reduces the concentration of the hydrogen ions in the bone matrix. Now, why is this important? Well, the bone mineralization happens in the alkaline environment. The idea here is that calcium is deposited in the bone matrix in case of alkaline environment. The reason for this is that the calcium and hydrogen ions always compete with each other for the binding sites on the proteins, either in the plasma, so they compete for the binding sites on the albumin, or in the bone matrix. They compete with each other for the binding sites on the bone matrix proteins. The less hydrogen ions we have on the bone matrix proteins, the more calcium can bind to those proteins. And deposition of calcium hydroxyapatite crystals in the bone is what's called mineralization. The source for osteoblasts is the stem cells present in the periosteum. Could you please remind me what the periosteum is? Exactly. The periosteum is the layer immediately around the cortical bone. In other words, immediately around the outer surface of the bone. And the periosteum contains the stem cells which can differentiate into osteoblasts and which can regenerate or rebuild the bone. For example, whenever the patient has a fracture, the periosteal stem cells will start to proliferate intensively so that they can produce osteoblasts, and then osteoblasts can rebuild the bone at the site of the fracture. Okay, and lastly, the question is, how do we measure the activity of osteoblasts? What are the markers for the activity of the osteoblasts? First, that's the bone ALP. I'm stressing that this is the bone subtype of the ALP because alkaline phosphatase is also produced by the biliary system. Nowadays, we can check the specific subtypes of the ALP. So we can check the bone subtype of the ALP, the biliary subtype, but there is one very clinical trick by which we can deduce where the ALP is coming from. Another enzyme made by the biliary tree is gamma glutamotransferase, or GGT. Now, if you see on the patient's labs that both ALP and GGT are high, now you know that that ALP is coming from the biliary tree. But now let's say that we see the patient labs and we see that ALP is high while GGT is normal. This suggests that the ALP is probably coming from the bone. Okay, ALP is one marker by which we can measure the osteoblastic activity, but the other marker is the osteocalcin. Osteocalcin is another protein made by the osteoblasts. And osteocalcin is made by osteoblasts whenever osteoblasts are active, whenever they are making the collagen. And the last and the third marker of increased osteoblastic activity is the measurement of propeptides of the type 1 procollagen. Let's remind ourselves from the cell biology that propeptides of the collagen are those three individual chains of the amino acids which finally make up the triple helix of the collagen. And those propeptides are also produced certainly by the osteoblasts. Now, if the propeptides are high in the plasma and the urine, it means that osteoblasts are working really hard to make the type 1 collagen. Now, let's move on to the osteoclasts. Osteoclasts dissolve the bone or they crush the bone. So, osteoclasts crush the bone. C in the word osteoclast for C in the word crushes. The way osteoclasts dissolve the bone is that they secrete the hydrogen ions and they also secrete the collagenases. Now let's take a step back and let's explain this process in a detail. The osteoclasts contain the enzyme, intracellular enzyme called carbonic anhydrase 2. And carbonic anhydrase 2 combines the carbon dioxide and the water into carbonic acid. Carbonic acid simultaneously breaks down into the hydrogen ion and the bicarbonate. Bicarbonate stays inside the cytosol of the osteoclast while hydrogen ion is secreted out into the bone matrix. Let's remember our discussion several minutes ago about the competition of hydrogen ions and the calcium ions for binding to the proteins. We said that calcium and hydrogen ions compete for the same binding site on the proteins. Now, if osteoclasts release the hydrogen ions, those extra hydrogen ions will displace the calcium from the bone matrix proteins. So the calcium will come out of the bone into the blood. This process is called demineralization of the bone. So after this process occurs, after calcium is displaced from the bone, now we have osteoid. Osteoid is the first bone that's formed by the osteoblasts. It's the bone without calcium. It's composed of mostly of the type 1 collagen. And now what's needed is to break down the collagen by collagenases. So after secreting the hydrogen ions, the osteoclasts secrete the collagenases which break down the collagen. And this is how osteoclasts break down the bone. So first they remove the calcium crystals in order to demineralize the bone, and then they remove the collagen from that demineralized bone. The source for the osteoclasts is the macrophages. Well, osteoclasts are the bone macrophages. The monocytes in the blood go into the bone and they form the osteoclast precursor, or the macrophage in this case. Now let's talk about how the osteoclast precursor becomes the activated osteoclast. Here we'll discuss a very high yield relationship of the osteoblasts and osteoclasts. There is the receptor called RANK on the surface of osteoclasts. RANK stands for receptor activator of NF-kappa B, or nuclear factor kappa B. And the RANK receptor is responsible for differentiating the osteoclast precursor into the mature osteoclast. And once the RANK is activated, several osteoclast precursors fuse with each other and they create the multinucleated giant cell, which we call the osteoclast. Now, do you guys know what activates the RANK receptor? Logically, it's RANK ligand, or RANKL. Importantly, the RANK ligand, or the RANKL, is located on the plasma membrane of osteoblasts. It means that in order for the osteoclast precursor to form the osteoblast first should bind to the RANK receptor on the osteoclast precursor through their rank ligand, and this interaction will stimulate the differentiation of the mature osteoclast. So now you can see that the major cell or the manager cell in the bone is actually osteoblast. Osteoblast builds up the bone, but at the same time it controls the osteoclast, and therefore osteoblasts control the bone resorption as well. And this concept is very high yield for us to understand what the Paget disease of the bone is all about. We won't go into any further details about the Paget disease because we'll talk about it extensively in the later episodes, but still, the idea is that normally osteoblasts are supposed to control osteoclasts through the interaction of the rank ligand and the rank receptor. The osteoblasts make another very important protein called osteoprotegerin. It's abbreviated as OPG. Osteoprotegerin is the decoy receptor for the rank ligand. In other words, it binds to rank ligand and it prevents the rank ligand from binding to the rank receptor. If rank ligand doesn't bind to the rank receptor on the osteoclast precursor, then osteoclast precursor will not form the mature osteoclast, and therefore there will be no bone resorption. To summarize, the osteoprotegerin protects the bone from resorption, and we have made use of this amazing mechanism by creating the monoclonal antibody called denosumab. Denosumab mimics osteoprotegerin. It mimics osteoprotegerin in a sense that denosumab binds to the rankle and does not allow the rankle to physically interact with the rank receptor. In this way, the osteoclasts cannot be activated and the bone cannot be resorbed. So, could you please tell me in which disease will we use the denosumab? I really hope that you're saying osteoporosis, the disease where we have excessive bone resorption and we need to limit the bone resorption as much as possible to maintain the current bone mineral density. Okay, so we have talked about the osteoprotegerin, the rankle, and the rank receptor. Now let's talk about two hormones which affect both osteoblasts and osteoclasts. The first hormone here is the parathyroid hormone, or PTH. Parathyroid hormone has its receptor only on osteoblasts. So the only cell that the PTH can directly affect is the osteoblast. And at the low intermittent levels, which is normal in all people, PTH activates the osteoblast to the degree that the osteoblast makes the bone. What I'm trying to say here is that at low normal levels, the PTH has anabolic effect on the bone. It helps us to build the bone. But when PTH is chronically and constantly high, let's say in case of primary hyperparathyroidism or secondary hyperparathyroidism, technically in all types of hyperparathyroidism, then PTH excessively activates the osteoblasts. And when PTH excessively activates the osteoblasts, now osteoblasts increase the expression of the rank ligand on their surface. The more rank ligand there is, the more rank receptors are activated on the osteoclasts. Therefore, excessive PTH causes excessive osteoclast activation through the osteoblasts. And then those osteoclasts break down the bone. This is how we get the disease called osteitis fibrosa cystica in the setting of the hyperparathyroidism. Okay, more on PTH in the endocrine series. Now let's move on to the second hormone, which is the estrogen. Estrogen can directly affect both osteoblasts and osteoclasts. In osteoblasts, estrogen inhibits the apoptosis of the osteoblasts. In contrast, estrogen activates the apoptosis of the osteoclasts. To say it simply, estrogen saves the osteoblasts and kills the osteoclasts. Now, what will be the net effect? Well, the bone will grow. we will be able to rebuild or build the bone because osteoblasts are still there while osteoclasts are dying. This is why estrogen is crucial for maintaining the normal bone mineral density. At the same time, estrogen is the hormone that causes ossification of the epiphyseal growth plates. Epiphyseal growth plates, let me remind you, are the cartilaginous growth plates located between the epiphyses and metaphyses of the long bones. And since the epiphyseal growth plate is the cartilage, it can increase the bone length so it can increase the bone length. And this is how the person increases in height. Estrogen causes ossification of the growth plates by stimulating the osteoblast, and therefore estrogen closes the growth plates. And once the estrogen closes the growth plates in both girls and boys, neither she or he can increase in height anymore. And then when the person has estrogen deficiency, then it means that osteoblasts can no longer survive. So as osteoblasts are dying, but osteoclasts are no longer dying. Again, estrogen normally induces apoptosis in the osteoclasts, meaning no estrogen or low estrogen means increased lifetime and life cycle of the osteoclasts. And this is how patients with osteoporosis patients with estrogen deficiency, either surgical estrogen deficiency after oophorectomy or postmenopausal estrogen deficiency, get osteoporosis. Osteoclasts get activated, they become more active than osteoblasts, and they degrade the bone matrix. Okay, so we have already finished up with talking about the cell biology of the bone. And we've come to an end of our today's episode, so let's summarize everything that we've discussed today. In this episode, we started discussing the types of the skeletal muscle fibers. We compared and contrasted the type 1 and type 2 skeletal muscle fibers, and we also pointed out the physical activities and exercises which increase the proportion of either type 1 or type 2 muscle fibers. Then we continued our discussion by talking about the skeletal muscle adaptations, specifically atrophy and hypertrophy, and we discussed what happens to the number of the myofibrils and myonuclei in both atrophy and hypertrophy. The next topic in our today's episode was the vascular smooth muscle contraction and relaxation. We linked contraction to the calcium influx in the cell, and we linked the relaxation to the nitrous oxide influx inside the cell. The next topic was the muscle proprioceptors, specifically the muscle spindles and the Golgi tendon organs. We discussed the stimuli for each of these muscle proprioceptors, and we also described the reflex arcs for each of them. Then we talked about the bone formation, explaining what endochondral and membranous ossification mean. And finally, we wrapped up our today's episode by discussing the cell biology of the bone. Here we talked about the function of osteoblasts, osteoclasts, their interaction through rank physical contact, and then we talked about two hormones, which is PTH and estrogen, and their effects on the bone. You can leave the voice message on this episode to let us know how we can improve our podcast for you. So thank you for your kind attention, Zeus Emuliers, and see you on the next episode.